Hello everyone, my name is Joanna Przybyl. I am an instructor in the Department of Pathology at Stanford University. I work in the group of Matt van der Rijn. Today I would like to share with you our preliminary results of the genome-wide DNA methylation profiling study of desmoid tumors. Desmoid tumor studies in van der Rijn lab are focused on identifying molecular markers associated with clinical behavior of these tumors. Specifically, our goal is to identify markers that could predict development of local recurrence after surgery and may help explain the differences in clinical outcome of tumors arising in different anatomical locations and carrying different mutations in the gene encoding beta-catenin. We previously performed a gene expression profiling study that showed that desmoid tumors show relatively homogeneous gene expression profiles. The candidate markers that we identified could not be validated by immunohistochemistry or in situ hybridization, which indicates that the differences in expression profiles between distinct groups of desmoid tumors may be fairly subtle. In recent years, it has been shown in many types of cancer that DNA methylation profile may provide valuable information for improved diagnosis or prediction of clinical behavior of different types of tumors. That's why, as the next step in our studies, we decided to look into DNA methylation patterns of desmoid tumors. We included 11 specimens of desmoid tumors in our large DNA methylation study of different types of soft tissue tumors. We profiled these specimens using EPIC microarrays manufactured by Illumina. In the unsupervised clustering analysis of this dataset, we observed that desmoid tumors formed a separate cluster with a distinct DNA methylation profile compared to leiomyosarcomas, leiomyomas, and gastrointestinal stromal tumors. The desmoid tumor cluster is indicated in the light blue color. We included 11 primary desmoid tumor specimens in this analysis, tumors from different locations and with different mutations in the gene encoding beta-catenin. 7 of 11 patients included in this analysis developed local recurrence after the surgery of the primary tumor. We performed differential methylation analysis to identify markers that would be specific for primary tumors that developed local recurrence compared to those that did not develop recurrence. We also compared DNA methylation profiles of tumors from the extremity versus those from the abdomen, and we also compared DNA methylation profiles of tumors with T41A mutation versus those with S45P mutation. For each comparison, we identified differentially methylated positions, single CPGs, and differentially methylated regions that may contain multiple CPGs. The numbers of differentially methylated positions and differentially methylated regions in each comparison are summarized in this table. We identified 35 differentially methylated regions between primary tumors that developed local recurrence compared to those that did not develop recurrence. Specifically, in primary tumors that recurred, we identified hypomethylation of promoter regions of 24 distinct genes. It is known that hypomethylation in the promoter regions may result in higher expression of target genes. And because we have RNA-seq data available for exactly the same specimens that were profiled by DNA methylation, we were able to look into this type of association between methylation and gene expression data. And for one gene that encodes ovarian carcinoma immunoreactive antigen-like protein, we observed possible correlation between hypomethylation in the promoter region and higher expression of this gene in primary tumor specimens that develop local recurrence. This gene may be biologically important, especially because it is known that it is involved in STAT3 activation in different types of tumors, and it is involved in JAK-STAT signaling. When we compared methylation profiles of primary tumors from the abdomen and from the extremities, we identified 78 differentially methylated CPGs. However, we did not observe any correlation with gene expression data for these results. when we compared methylation profiles of primary tumors with T41A mutation and those with S45P mutation, we identified 25 differentially methylated regions. And interestingly, in tumors with S45P mutation, we identified hypomethylation of the promoter region of HOXA5 transcription factor. And we observed possible correlation between methylation status and higher expression of this gene in the same tumor specimens. These results look promising. They point to the fact that DNA methylation profiles may be associated with clinical behavior, tumor location, or different mutations in the gene encoding beta-catenin. And for selected genes, we also observe possible association between DNA methylation profile and RNA expression level. This was just a small pilot study based only on 11 samples, but based on these encouraging results, we propose that a larger DNA methylation study may allow for a discovery of clinically relevant markers associated with clinical behavior of desmoid tumors, and integrated analysis of DNA methylation and RNA-seq data in a larger dataset may reveal new regulatory mechanisms underlying desmoid tumors. I would like to acknowledge the members of Matt van der Rijn's lab that helped to perform this study, and I would like to also acknowledge the bioinformatics group led by Benjamin Heibeck-Heinz from Toronto and Chris Ellis from his group, with whom we jointly developed the computational pipeline for the analysis of DNA methylation data. Thank you very much for your attention.